Welcome, dear listener, to Episode 7. Today, I'll introduce the cell and cellular functions, but first, a little housekeeping. The last 5 or 6 episodes were part of a series about the classes of biomolecules that make life work. This episode is the first in another series, which will cover the cell and its physiology. Where I started the podcast by exploring biomolecules, I'm now zooming out the microscope to explore the next level of chemical organization in living things. After this series, I'll make more that will explore higher and higher levels of organization. Think body systems, individual organisms, populations, and whole ecologies. But before I can even cover any of that other stuff, I have to know that you, the listeners, know what I'm talking about. That's why I have to start small, with molecules and cells, and work my way up. Alright, now that that's out of the way, let's get started. Cells are quite massive compared to molecules. An enormous bubble of phospholipids studded with proteins forms the cellular membrane, while many more proteins, carbohydrates, and nucleic acids are packed into the cell itself. There are 2 major domains of cells, which I referenced in the first episode of the podcast. These 2 domains are eukaryotes, like humans and fish and birch trees, and prokaryotes, which are very small single-celled organisms. The domain prokaryote has 2 major divisions, archaea and bacteria. I'll discuss more about these later in the episode. It's currently accepted that the first protocells evolved into prokaryotes, which gradually became more sophisticated until the eukaryote lineage branched off sometime later. This chronological series of events is logical, as eukaryotes are both larger and far more complex than prokaryotes. However, it should be understood that the issue is far from settled. We really aren't sure what the first cells were like, and there's a lot of controversy over the data. Some people posit the argument that the evolutionary order is backwards. Eukaryotes were the original cell lineage, but a subpopulation evolved towards simplicity and branched off to become the prokaryotes. In 1998, a scientific paper about the universal ancestor of all life put forth the argument that the 3 domains, archaea, bacteria, and eukarya, emerged during roughly the same time period from within the same gene pool. We cannot be precisely sure which came first, although this isn't to say that some revelatory discovery in the future can't put the matter to rest. The primary physical differences between eukaryotes and prokaryotes are their internal organization and their method of DNA storage. Prokaryotes keep their DNA in the shape of a large loop, free-floating in their cytoplasm. In fact, everything in the prokaryote floats in the cytoplasm, as they don't possess internal membrane-bound compartments. Eukaryotes keep their DNA inside a nucleus, which I'll discuss in more detail in a few minutes. Eukaryotes also have numerous internal compartments called organelles, membrane-bound spaces that hold specific chemicals or perform certain functions. I'll also discuss these in more detail in a few minutes, but to start things off, I'm going to explain the structures that are common in both eukaryotic and prokaryotic cells. All cells have a plasma membrane, a very important lipid bilayer membrane. This membrane is studded on both sides with a huge collection of proteins and glycoproteins and other types of molecular structures. Together, these proteins work to strictly regulate what does and does not enter the cell. In this way, the membrane creates a hard division in space between live matter and non-living matter. That which is on the outside is the non-living external world. That which is encapsulated by the cellular membrane, all the enzymatic machinery and biomolecules and ions, this is what composes living matter. All cells also have a cytoskeleton of some kind. This cytoskeleton is usually composed of many thin filaments, like fibers or coiled tubes. They're layered across the inside of the membrane, and they reach across through the cytoplasm. They're physically connecting the internal sides of the cell. The cytoskeleton provides a measure of stability and consistent structure to the otherwise fluid and amorphous membrane. During cellular replication, the cytoskeleton assists in pulling apart the cytoplasm and orchestrating a clean division of the membrane. In prokaryotes, diffusion can move most nutrients and resources through the cytoplasm quickly. In larger eukaryotic cells, diffusion is less effective, so proteins and protein complexes must be used to move stuff around inside the cell. These proteins moving within the cell use the filaments of the cytoskeleton like roads or bridges, crisscrossing through 3D space, to move items to different regions inside the cell. Some types of cells in both the eukaryotic and prokaryotic domains have a cell wall. Prokaryotic bacteria and eukaryotic plants both have this cell wall feature, which is composed of a type of carbohydrate, or in some cases, a polysaccharide. and this is deposited on the outside of the membrane. So the cell builds this by oozing out the material out of its membrane, and that deposited material, forms a solid, rigid crust on the outside of the membrane. So the cell wall, it's rigid, and it provides a structural integrity and a mechanical protection. When the solute concentration of the cell is high, water will come into the cell through osmosis, causing the cell to swell and expand. In extreme cases, the cell will burst and spill its guts out into the extracellular matrix, or into the solution, or into the water, whatever happens to be. The cell wall is a defensive measure against this. Its rigid encapsulating structure prevents the membrane from swelling past a particular size. The pressure within a prokaryote, for example, is nearly the same as the pressure in a car tire. Imagine that. Some prokaryotes also share with plants the capacity for photosynthesis. The chemical processes of capturing the energy in sunlight was one of the first major evolutionary shifts in the history of life. As a result, many prokaryotic species are photosynthetic, producing sugars through the enzyme-dependent process of photosynthesis. I'll be doing a future episode about photosynthesis when I'm discussing plants in greater detail. All cells have genetic material, which they reproduce during cellular division so that each daughter cell has its own copy of the parent genome. If you listen to the first episode, you might recognize a lot of this material from the formal requirements for life, or from cell theory. Other parts of cell theory discuss reproduction, of both DNA and the cells themselves. Every prokaryote species is unicellular and produces asexually. They divide themselves to produce two daughter cells. Eukaryotic species are unicellular and multicellular, and they can reproduce asexually or sexually. Sexual reproduction involves the combining of two individual gametes into a new genome. Every species has DNA. These long strands of nucleic acid coil together tightly into a supercoiled rope-like structure called a chromosome. In both prokaryotic and eukaryotic cells, chromosomes are also composed of proteins, which help organize the coiling and packing of the DNA strands. The structure and location of the chromosomes within the cell, however, differs between eukaryotes and prokaryotes. I'll discuss these differences in chromosome structure in a few minutes as well. Consider the proteins that transcribe and translate DNA. These proteins use a complex structure called a ribosome to finalize the production of proteins. The ribosome has a large and small subunit, each one consisting of both proteins and RNA molecules. Together, these subunits work to connect amino acid codons into polypeptide chains, which then get pushed out of the ribosome complex one monomer at a time. The final product is a polypeptide chain that naturally folds itself into a functional protein. Cells have thousands of these ribosomes, constantly printing out proteins according to the cell's needs. In this way, the information stored in the genetic code gets chemically transformed, ultimately coming to a ribosome which prints out a protein. The specific A, T, C, G sequences of a gene code for a specific protein, and the proteins do the functions that maintain life. If you think that's cool, just wait. There'll be a whole series about genetics that will dive into the details of this process. Now that I've covered some of the similarities between all cells, I'm going to discuss the differences between eukaryotes and prokaryotes. Perhaps the most striking difference between the two is their size. Prokaryotic cells are much, much smaller than eukaryotic cells. So much so that eukaryotic cells often entirely consume prokaryotic cells as a manner of lunch. The smaller size of the prokaryotic cell gives it a larger surface-to-volume ratio. In biology, a critically important rule that you should always keep in mind is that form implies function. The function of a molecule or cell or whatever is dependent or heavily influenced on its structure. In this case, the larger surface-to-volume ratio allows prokaryotes to absorb chemical resources through its membrane at a faster rate. What about the structure and organization of the chromosome in the prokaryotic cell? The prokaryotic chromosome takes the shape of a giant loop, free-floating in the cytoplasm alongside all the other internal contents of the cell. The chromosome is very long, coiling and piling back on itself in a loose clump somewhere within the cell. The location of this tangled mess within the cell is called the nucleoid. Particular segments of the chromosome are organized into loops. Regions within these loops can selectively decompress, allowing the DNA to kind of unfold a little bit and loosen slightly, and this allows proteins and enzymes to access the DNA strand and begin to transcribe particular genes. Besides the nucleoid, prokaryotes like bacteria can keep genetic information in much smaller loops of supercoiled DNA, called plasmids. The plasmids provide genes that help the prokaryote react to rapid shifts in its chemical environment, like a sudden change in acidity or temperature. Prokaryotes can also do something called horizontal gene transfer, where one or more genes is replicated in one cell and transferred to another cell through one of three processes: bacteriophage-mediated transduction, plasmid-mediated conjugation, and natural transformation. In transduction, a virus-like bacteriophage carries the genes, and it infects the other cell with them, kind of like how a virus or bacteria would infect your own cells. In conjugation, the two cells physically touch each other, either through membrane contact or through a thin bridge-like structure. It's like a tube that they connect to each other with. Through this, they share genetic information. This is kind of like bacteria sex, or the closest thing to it at least. The third process of horizontal gene transfer is called transformation, where the cell absorbs a free-floating piece of genetic material and incorporates it into its own genome. All but two genera of prokaryotes have a cell wall, a stiff sheet wrapping around the membrane. Of the two domains of prokaryotes, bacteria and archaea, one of the major differences between them is the composition of their cell walls. These cell walls come in a variety of different shapes, which can be helpful in organizing and classifying species. Bacteria and archaea can be spherical, pyramidal, or cone-like, rod-shaped, or in the form of a short helix or spiral. Bacterial cell walls are composed of the carbohydrate peptidoglycan. Some bacteria have thick, heavy peptidoglycan shells, which stain a dark purple under the Gram stain test. When seen through a microscope, bacteria with thick cell walls appear as dark purple blobs. These bacteria are gram-positive, denoting the thick layer of sugars coating their membrane. Gram-negative bacteria, in contrast, have thin cell walls that stain lightly under the Gram stain test. Under a microscope, gram-negative bacteria appear as a very pale purple or a barely visible pink. There are 4 types of archaean cell wall, each composed of a different material. The first type of archaean cell wall is called Pseudo Peptidoglycan. As its name suggests, it's similar to peptidoglycan, but it has a beta-1,3 linkage instead of the beta-1,4 linkage that's found in peptidoglycan. So the second type of material is composed of a mixture of glycoproteins and methanogens. The third type is it's a single layer film of proteins called an S layer. The final type of cell wall is poorly understood, but it's known to be composed of a thick layer of polysaccharides. Furthermore, prokaryotes often have an array of molecular structures protruding from their membranes. You might be familiar with something called a flagella. Many bacteria have a flagella, which are long, string-like structures with a corkscrew shape. Enzymatic engines residing in and underneath the membrane are able to rotate the base of the flagella, and this causes it to spin. The spinning propels the prokaryote through the water at speeds much faster than any fish can go relative to their body size. Some bacteria also possess numerous needle-like structures protruding from all over their membranes. These needle structures are called fimbria, and bacteria use them as an anchor, or like a pin to pierce themselves in. These fimbria are literally used to stab into the membranes of other cells. Many infectious bacteria owe their persistence to the fimbria, which stick into host cells where they become really difficult to remove. So that about covers the unique characteristics of prokaryotes. Recall how much larger eukaryotes are than prokaryotes. Their large size gives eukaryotes a larger surface-to-volume ratio. They have a slower metabolism, and they take longer to grow and reproduce. But they also have a greater level of internal organization, which promotes the efficient use of resources and allows for a greater degree of functional variation. Eukaryotes build this internal organization with a number of smaller internal compartments. They're smaller compartment-like bubbles of bilipid membranes within the cell's main membrane. This compartmentalization also increases the effectiveness of diffusion. Diffusion is less effective as a method of transport over long distances, like the full length of a eukaryotic cell. But by breaking up their internal space into smaller compartments, the effectiveness of diffusion increases. Resources can be distributed via diffusion throughout the compartment much faster than they could through the whole cell. So the bubble compartments can create isolated chemical environments, One compartment can isolate the synthesis of a particular biomolecule, while another compartment can contain chemicals that break down and recycle that biomolecule. So furthermore, by isolating the chemical environments, compartmentalization also purifies these environments. Necessary chemicals can be held in high concentrations. Similarly, necessary molecular machinery like proteins and enzymes and stuff can also be produced locally and kept nearby. Resources can be easily resupplied through diffusion. The advantages are numerous. I've already mentioned the most significant compartment within eukaryotic cells that prokaryotic cells lack. This is the nucleus. The nucleus is typically the largest organelle, and it's composed of a double bilayer membrane called the nuclear envelope, which is pierced by a collection of highly regulated pores. The nucleus has a cytoskeleton of its own, composed of protein filaments called nuclear lamina, and these line the interior surface and they provide structural integrity. Within the nucleus exist two types of structures, the chromosomes and the nucleolus. The nucleolus is a contiguous region, appearing under the microscope like a dark mass. It's here in the nucleolus where ribosomal RNA is synthesized and the subunits assembled. The chromosomes, on the other hand, are loosely organized, unpacked, each one floating in a particular section of the nucleus. During cellular replication, the chromosomes condense into supercoils. Surrounding the nucleus is the endoplasmic reticulum, or the ER. The ER is a large, flattened organelle with tubules and pockets, all kind of interconnected and flowing back and forth between one another. It's one in a continuous membrane, but its folds make it appear, in transmission electron micrograph photos to look like ripples or droplets surrounding the nucleus. The ER is a busy place. A lot of stuff happens here. To make it simpler, the ER can be divided into two distinct regions: the rough endoplasmic reticulum, called the ER, and the smooth endoplasmic reticulum, or the smooth ER. So rough ER and smooth ER. The difference is that the rough ER is covered in ribosomes. These ribosomes give the membrane of the rough ER a studded, gritty appearance, hence the name rough ER. Proteins are constructed by these ribosomes, which eject them into the lumen, or the internal cavity of the rough ER. Here, the proteins can fold themselves properly and they get organized according to their destination in the cell. You see, the rough ER doesn't just act as a place for proteins to fold safely. It also packages these proteins in smaller compartments that it buds off of its main membrane. These budded membrane pockets are given chemical tags, which tell other proteins in the cell where the membrane should go. In this way, particular enzymes can be produced and sent to the organelles that need them. The smooth ER, in contrast, has no ribosomes, and thus appears smooth. The smooth ER is where the cell synthesizes and breaks down lipids, where metabolic byproducts are detoxified, and where carbohydrates and steroids are metabolized. Steroids are also one of the lipids produced by the smooth ER. In steroid-producing tissues like the gonads, cells are characterized by a significantly higher volume of smooth ER than exists in the cells of other tissues. So this high volume of smooth ER is necessary for the specialized production of steroid hormones like testosterone and estrogen. The smooth ER also produces the phospholipids used in your cellular membranes. Without a continual replenishment of the phospholipids, your membranes would gradually decay and weaken. Like the rough ER, the smooth ER also packages its products in little bubbles, and these bud off from its main membrane, and they send them where they need to go. The rough ER often sends its little membrane packages directly to the Golgi Complex, another large organelle that acts like a post office for the inside of the cell. The Golgi apparatus is not just one membrane, but it's instead a number of flattened membranes called cisternae, and they're stacked on top of each other like pancakes. One flat side of the Golgi apparatus faces the nucleus, while the other flat side faces the cell's main plasma membrane. The side that faces the nucleus is called the cis side, while the side that faces the plasma membrane is the trans side. The Golgi apparatus receives protein-filled vesicles from the rough ER on the cis side, and these are absorbed into the cisterna and processed. The enzymes of the Golgi apparatus are anchored to its membranes, so this is where the bulk of the processing happens, along the membrane, or right next to the membrane. Various cisterna are responsible for particular acts of protein modification and processing. So, for example, the cisterna that are near the cis side help to remove a sugar called mannose. Cisterna in the middle do that, but they also add a monosaccharide tag called N-acetylglucosamine. Cisterna on the trans side add galactose molecules to the proteins. These are just examples. The Golgi complex is capable of creating a huge variety of modifications to suit just as many of the cell's needs. The proteins are shuttled through the Golgi complex, through successive layers of cisterna, until they bud off the trans side and float to their destination. Animal cells possess lysosomes. If the Golgi apparatus can be likened to a post office, the lysosomes can be likened to a trash recycling plant. The inside of the lysosome is a very acidic place, containing a solution with a pH of 5 or 6, so this is acidic relative to the rest of the cell. The acidic conditions are maintained by proton pumps, and these proton pumps lower the pH by increasing the concentration of protons or hydrogen ions within the lysosome. So these little acid bubbles float around the inside of your cells, a perfect example of the isolating capabilities of a plasma membrane. Old and degraded macromolecules, like proteins, nucleic acids, carbohydrates, and lipids, are broken down through a series of condensation reactions. Eukaryotes also have peroxisomes. These are spherical organelles which emerge into the cell as buds coming off of the endoplasmic reticulum. The peroxisomes are responsible for isolating reduction-oxidation reactions. It's good that the peroxisomes effectively contain these caustic reactions, because the chemicals involved can destroy most other biomolecules. So, for example, some compounds produced in the peroxisomes are used to generate bile acids. These bile acids are part of the digestive system. They allow the body to dissolve and absorb fat-soluble items like lipids and vitamin A. In the leaves of plants, cells possess a type of peroxisome called the glyoxysome. This leaf-dwelling peroxisome oxidizes fat molecules and converts them to an energy storage molecule. This makes sense, as the leaves are an energy-producing tissue, so they would produce energy-storing molecules, or just molecular energy. Seeds, on the other hand, have a different type of peroxisome with a different suite of enzymes. These seed-dwelling peroxisomes break down energy-storing molecules to feed their initial growth. In other cells, these oxidative enzymes break down fats and feed the pieces to the mitochondria. The mitochondria then makes energy. So the mitochondria themselves are one of two types of organelles that exist in eukaryotic cells through an endosymbiotic relationship, the other organelle being chloroplasts, which I'll discuss momentarily. So both mitochondria and chloroplasts exhibit unusual behavior for an organelle. They have their own genome of bacteria-like DNA, separate from the cell's genome, and they divide at their own pace, not in accordance with the cell's division. These observations led to the formation of the Endosymbiosis Theory, which posits that mitochondria and chloroplasts were once free-floating bacteria. At some point, eukaryotic cells would have ingested these bacteria, but instead of destroying them, they incorporated them into their own physiology. This would only occur if the bacteria and the cell could benefit from one another. So the cell produces the foods needed for the bacteria, and the bacteria produces energy for the cell. The mitochondria produce the bulk of the cell's ATP, which is why it's referred to as the powerhouse of the cell. 50 to 1,000,000 mitochondria per cell produce the chemical energy used to power virtually everything the cell does. Like the nuclear envelope, the mitochondria have two layers. The inner layer of the mitochondria, however, doesn't line the outer layer perfectly like it does in the nuclear envelope. The inner layer of the mitochondria, it folds back and forth on itself numerous times, filling the space of the organelle with sheet-like folds. So it's kind of like if you have a plastic bag, that's one size, and you have a second plastic bag that's much larger, it's got a much larger surface area, and you crumple up the larger bag and stick it into the smaller bag. That's kind of what the outer and inner membranes of the mitochondria are like. The intermembrane space between the inner and outer membranes forms pockets within these sheet-like folds called cristea. A fluid kept within the inner membrane is the mitochondrial matrix. Enzymes sit all over the inner membrane and float through the matrix, harvesting energy from the food molecules to produce ATP. A typical cell will have enough mitochondria to produce nearly 10 million molecules of ATP a second. Muscle cells use even more. This is because muscle cells are very large. They stretch from one joint to another. And for a muscle cell to work, you need to have a massive number of contracting motions on a molecular level in every single cell. This consumes an absurd amount of ATP. And so muscle cells and other, high energy cells or cells that use a lot of energy, they need to produce a lot of energy. Plant and algae, they have cells that possess both mitochondria and chloroplasts. Like mitochondria, the chloroplasts have an outer and inner membrane. The inner membrane of the chloroplast, however, is not folded into sheets. Instead, they're broken up into hundreds of individual disc-like bubbles. These membrane-bound disc-like bubbles are very flat, and they stack together like pancakes. I know I've said that a lot in this podcast, but this is how this stuff is. They all stack together, and they're like pancakes. And so these stack of membrane-bound bubbles, they're separated from the outer membrane by a space called the stroma. And so each flat bubble, each little pancake, is called a thylakoid. And the stacks they form are called grana, or granum, or granum, I guess. Granum is probably the correct pronunciation. It's within these thylakoids that proteins derive energy from photosynthesis. Enzymes within the stroma use this energy to build sugars, which are used to grow the plant's tissues. A final type of organelle, called a vacuole, exists in both plant and fungal cells. The vacuole is very large, in some cases taking up 80% of the cell's internal space. The organelle is so large because its high concentration of sodium and potassium ions causes water to flood into it through osmosis. The vacuole is a storage site for all manner of molecules and ions. In seeds, the vacuole is filled with proteins. These proteins are like a giant amino acid conglomerate, and it's digested by the young plant, or the seed, for the raw amino acids that it needs to build its own proteins. The cells in the stems and leaves have vacuoles filled with unpleasant compounds that discourage animals from eating it. Some vacuoles are filled with tannins, a group of molecules that taste unpleasantly bitter. Drug-producing plants like coca or the coffee bean accumulate the drug compound in their vacuoles. This compound, stored in a particular cell's vacuole, can play a role in that cell's function or specialization. And all cells are really specialized like this in some way. Groups of cells that share a similar specialization, or a suite of specializations, they come together to become tissues, and these various tissues compose the organs and flesh of an organism. So all cells have a variable structure and a variable proportion of organelles depending on their function. Muscles, for example, have long fibrous cells filled with parallel protein filaments. The protein filaments crawl past one another when the cell contracts. And so when many muscle cells contract at once, the muscle itself contracts and limb movement is created. This contraction requires a lot of energy. So to power all of this movement, the muscle cells have a relatively high number of mitochondria. The parietal cells in your stomach, for example, are also full of mitochondria. These parietal cells are the guys responsible for producing your stomach acid. And so producing stomach acid is a pretty energy-expensive process. And having more mitochondria ultimately makes for a more productive parietal cell. Consider the hormone-producing cells in the gonads, namely the Leydig cell and the granulosa cell. These cells use cholesterol molecules as precursors for other steroid hormones. To be specific, the Leydig cells in the testicle produces testosterone, and the granulosa cells in the ovary produces estrogen. The Leydig and granulosa cells have a lot of smooth endoplasmic reticulum, and this is because the steroid molecules that they produce are lipids, and the smooth ER produces and synthesizes lipids. And so it just makes sense that the cell that's specialized to produce lipids has a lot of the internal organelles, dedicated to producing lipids. Form follows function. The body has endless examples of cells whose form facilitates their function. So consider your neurons, whose long axons and branching dendrites allow hundreds of them to connect together, enabling you to think complex thoughts and reflect on your life. Consider the lining of your intestines. Enterocytes develop thousands of microvilli along their apical side. These microvilli are like thin tendrils, and together they increase the surface area of the enterocyte tremendously, allowing it to absorb more nutrients. So consider the cells that produce the mucus in your nose with their goblet-like structures, or the cells in your eye with their light-absorbent pigments, or the thin, slippery cells lining the inside of your veins. All cells have some kind of specialization. When large groups of cells all have the same specialization, they form tissues. These tissues do the things that living organisms need in order to survive. Your muscle tissue, for example, moves your limbs by applying forces to your skeletal tissue. Your neural tissue is responsible for both conscious thought and the unconscious regulation of body systems like your heart rate, body temperature, and the depth of your respiration. Your stomach tissue produces acid and other compounds that break down food. Your dermal tissue acts like a hairy spacesuit to keep out pathogens and protect your soft innards from ultraviolet radiation. That's about it for this episode. I think it's a pretty good introduction to the cell. Hopefully you thought it was too. Hopefully you learned something neat. This episode is the first in a series covering the cell and its functions. Next episode, I'll be discussing the subsystems of the cell in greater detail, including the internal transport mechanisms and the cytoskeleton. If that sounds interesting, stick around so you can check it out. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogic Podcast. To buy something from the store, go to redbubble.com and search for BioLogic, or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/biologicpodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.